Okay, so just to introduce myself first, I'm Claire Anderson and I'm part of the scientific communications team. I actually first discovered Unique just over 10 years ago now when my niece was diagnosed with tetrasomy 9p, and my sister had what I'm sure will be quite a familiar experience to a lot of you, with quite a traumatic diagnosis. She came away not really knowing what this might mean for Tilly and for the family. She had no idea really what to expect, and she wasn't signposted to anywhere. So luckily, a paediatrician friend of mine, Ruth, said get in contact with Unique and they might have some families you could connect with or some information, and they did. So that made a real difference. So I have a background in, my degrees in human genetics, and then I have a PhD in immunology, and I worked as a research scientist in vaccines. So I, first of all, volunteered for Unique, helping write guides, and then since 2017, I've been part of the guide writing team.
Perfect. I think I'm next. Yes. Hello, I'm Neve Marshall. So I actually straddle both the guide writing team and the helpline team. So for the helpline team, we sort of do the emotional support and, answering your guys' questions through email or telephone or, however you guys want to get in touch with us. And then the Unique guides as well, as I write a few of those too. My background is I am a registered genetic counsellor, recently registered, and I also taught science in a special needs school. So got lots of experience working with lots of different type of people. And yeah, that's me.
Hi, I'm Emily. So I actually came to studying genetics a little bit later in life, and that was because of my own family's experiences. So my daughter's got a couple of rare conditions. There may or may not be a genetic cause. We're probably going to have some more testing done. But my husband's family have also got a genetic condition. So there was a period of time when I was learning lots about genetics, and I thought, you know what, this is actually really interesting. So a few years down the line, I've now got a PG cert in genomic medicine. I'm in my third year of a master's in genetic counselling, and I'm very happy to be part of the Unique team on the helpline team with Neve and some of the others.
Oh, is that me? Right. Gonna try and click and speak at the same time. So we're going to do a little brief background into sort of genetics and some of the key aspects of genetics. Again, any questions at the end, you can get them this way. Right. So essentially, we're all made up of billions of cells, and inside each of our cells we have our genetic information called DNA. DNA is like a long instruction manual made up of a 4-letter code, A, T, G, and C. So if we imagine it a bit like a recipe book, who's it, Nigella Lawson? She's got lots of different types of books. We can imagine those like our chromosomes, so lots of different books, and then inside those we have individual recipes, which we call our genes. So yeah, along our DNA are sections called genes. And then each gene provides the body with specific instructions, such as to grow a heart or organs, how the brain works and grows, and how we function as well. So our DNA is packaged into tightly coiled structures called chromosomes. We have 2 pairs of chromosomes, and that's because we get one from mum and one from dad. So we normally have 46 arranged into 23 pairs. The 23rd pair is our sex chromosome. So women are XX and men are XY. Chromosomes can be stained and viewed under a microscope. So that's what our scientists do when they start looking into chromosomes to see, whether someone has a change in their chromosomes and things like that. And we have our P arm of the chromosome, which is the shorter arm, and the Q arm, which is our longer arm as well. And the numbering that you sometimes see when you have a genetic test back refers to the numbers going outwards from the centromere, and that's the centromere in the middle. Passing on.
Thank you, Neve. So now we've learned a little bit about genes and chromosomes. I'm going to talk a bit about the ways that genetic information can be changed. So sometimes, instead of having the expected amount of genetic material or DNA, there can be missing or extra pieces. So these might either be deletions or duplications, and you might sometimes hear them referred to as copy number variants or CNVs. So with a deletion, which is shown on the left-hand side of the slide, that's where there's a piece of the genetic material missing. So Neve's used the example of a recipe book. I guess you could also think of it as like an instruction manual for the body. So with a deletion, some of those instructions are missing, so they're not there for the body to use. Whereas with a duplication, which is on the other side, you've got extra duplicated or copied genetic information. So there's more instructions there for the body to use than there would otherwise be. So hopefully you can see that shown there on the diagrams. So deletions and duplications are measured in base pairs. So the numbering that Neve was talking about. So deletions or duplications can be very small. They can be much larger. They might involve lots of genes or even whole chromosomes. And the way that a deletion or duplication affects somebody can depend on the size of the change. It could depend on what genes are there, what kind of job they do. But there's also lots of other factors that are involved in how a deletion or duplication affects someone, if at all, and some of those we don't fully understand yet. So these kind of changes can happen for the first time in somebody, which is called de novo, or they could be inherited from a parent. So that's what happens when there's a difference in the amount of genetic material. But there are also situations when the amount of genetic information is the same. It might be just rearranged in a different way. So one example of this is a balanced reciprocal translocation, which is shown on the PowerPoint. So this is where a section of one chromosome is broken off and a section of another chromosome is broken off, and they've essentially swapped places. So going back to the instruction manual, it's a bit like imagining that two of the recipes or two of the chapters or instructions have swapped places, but they're still there. So all of the genetic information is still there for the body to use. So most of the time, not always, but most of the time, someone with a balanced translocation doesn't have any features or symptoms of a condition themselves. They might not know they have it, although it can sometimes cause recurrent miscarriage or other kind of issues with fertility. So although usually someone's health isn't affected by having a balanced translocation, it can become important when thinking about having children, because there's different ways that those chromosomes might be passed on, but one possible outcome is that an unbalanced rearrangement might be passed on to a child. And so this is what we would call an unbalanced translocation. So in this case, the genetic information or the instructions have been rearranged, but in a way that there is some genetic information missing or added or both. So the way that that might affect someone can depend on what kind of chromosomes are involved, what genes are present. Sometimes this can lead to pregnancy loss, or it can sometimes also lead to problems of health and development with the child as well. And this is something that can happen if someone has a balanced translocation, but it can also, like the others, happen for the first time in a child. There's also a particular kind of translocation that behaves a little bit differently, and this is what we call a Robertsonian translocation. And these kinds of translocations only happen with particular chromosomes, and those are chromosomes that have a very short, a short P arm or a short arm. So a lot of the information that's contained on those chromosomes, the short arm of those chromosomes, is kind of found elsewhere. So when a Robertsonian translocation happens, you can see you kind of lose these short arms from two chromosomes, and then the long arms then join together and fuse to make one chromosome. So even though the short arms have been lost, the central kind of genetic information is still there for the person. So this person would have a balanced Robertsonian translocation, sometimes known as a Robertsonian translocation carrier. And again, usually that wouldn't affect their health, but it can affect the way that chromosomes are passed down to children, and that can cause conditions like Down syndrome and some other conditions, for example. Oh, sorry. No. Sorry, we're having a technical... I don't think it's going to let me go back. So unfortunately, I can't show you what a single gene condition is. Maybe you saw it quickly. But those are the kind of situations where we're talking about pieces of DNA. But actually, sometimes people can have a change in a single gene, so a very small kind of change. And we've all got lots of changes between us. And sometimes, especially these really small kind of changes, and sometimes they don't have any obvious effect. But because genes are responsible for building something that the body needs, usually a protein, if that change affects whether the protein's made or how it works, then that can cause a genetic condition. And there's different ways, different kind of patterns of inheritance, depending on what gene is involved. Again, though, like with the others, this is something that can happen for a child for the first time. So genetics can tell us a bit about how things happen, but they don't always tell us what effect it will have on a person, which I think Claire will talk a little bit about in a moment as well. Thank you.
Yeah, we're almost there, just a little bit more information. So I think when most of us think about mosaics, it's probably because we've visited a lovely Roman ruin and seen mosaics on the floor in sort of tiles, or in art classes where we may have made them. So in those circumstances, a mosaic is made up of small individual pieces of coloured material which come together to form a pattern or image. And there's a lovely, beautiful double helix there, which I'm sure the Romans would have had on their floors had they known about it at the time, but they didn't. But in terms of genetics, when we're talking about mosaicism, it describes a condition in which a genetic change is found in some, but not all, of the body's cells. And that's really what we want to get across. So some cells have the genetic change, but not all of them. And there are two types, somatic and germline, and it's possible to have one or the other or both. So when we talk about somatic mosaicism, a somatic cell is any cell in our body other than egg or sperm cells. So when we have a somatic mosaicism, it refers to a genetic change that is found in some of the cells of the body, but not in the egg or sperm cells. And again, it's only found in some of the other cells in the body. So if it happens, this genetic change occurs early in development, then it's more likely that a greater number of cells, so a large proportion of cells in the body, will be affected, and this is called constitutional mosaicism. Whereas if it happens later in our lives, then it's more likely that only a specific area or a specific organ will be affected and carry that change, and that's called confined mosaicism. So if a person with somatic mosaicism, they may or may not be affected by having that genetic change, depending on how many and which cells carry the change. And crucially, it's not passed on to the children because it's not in the egg or sperm cells. And then the other type of mosaicism is germline mosaicism, and this is rarer than somatic mosaicism, and it refers to a genetic change in some of a person's egg or sperm cells, so not in the somatic cells. So a person with a germline mosaicism will not be affected, but they can pass that variant on to a child, who is then likely to be affected or have some of the health or developmental concerns associated with the genetic condition that that genetic change causes. So if someone has germline mosaicism, they may well know nothing about it until they experience one or more pregnancy or have one or more child affected by a genetic change. And it wouldn't be picked up in standard genetic tests. And crucially, each pregnancy carries a chance, a variable chance of passing on that change to the child. And then last two slides, and I think this kind of brings everything together, and it actually is summed up in our name, unique. We are all unique, and actually that's also reflected in how we respond to a genetic change. And that can be even within the same family, and it's something that when I started working for unique, took me a long time to get into my head that it really is very hard to predict what a particular genetic change might mean for a person. So we use two terms for this, penetrance. So penetrance is used to describe the proportion of individuals with a particular genetic change who exhibit any sign or symptom of that genetic disorder. And if something has complete penetrance, everyone who has that genetic change will be affected. So you can see everyone's coloured in there. It's got 100% penetrance. Whereas if we have reduced, or sometimes called incomplete penetrance, that means some individuals will show features or symptoms, but others won't display any obvious features or symptoms. So here you could see that 7 out of 10, and we would say 70%, so that condition has 70% penetrance. So it's kind of an all or nothing binary type thing. But there's also expressivity that we have to consider. And expressivity describes how different individuals may experience a range of features and symptoms with differing severity despite having the same or similar genetic change. So we talk about low expressivity, and that means the majority of individuals, as illustrated here, they have the same or similar genetic change and very similar features and similar severity. However, if we have something with high expressivity, that means there's a broad range of features, a broad spectrum, and also a broad range of severity in people who have the same or similar genetic change. So expressivity is more like a spectrum. Penetrance is more binary. This is more of a spectrum. And it's this that makes genetic counselling really quite tricky. We know that most genetic disorders have variable expressivity and some have reduced penetrance. So if we look at the bottom slide here, if we had complete penetrance, so that's 100% of people would be affected, but variable expressivity, it means everyone's affected but in different ways. Whereas if you have reduced penetrance and variable expressivity, you'll have some people have no symptoms or features seemingly at all, whereas others are affected but in different ways. And as Emily alluded to, we don't really totally understand why this is. But we do know that for many genetic conditions, the onset of symptoms is related to things like age, sex, genetic background. We all have our own unique genetic background. If you've got additional genetic changes, variations in gene activity levels, and other environmental factors that we are only just beginning to understand. And the key takeaway from this, and it's very frustrating, but it is very difficult to exactly predict from a genetic change, from a genotype, what your phenotype, so what the expression, what that will mean for the person will be. It's quite complicated, so I would signpost you to our website where we have lots of guides going into these, not just our CNV guides that Niamh was talking about and Emily. We have single gene disorder guides, have general genetics guides talking about things like mosaicism and penetrance. We have practical guides to things like everyday living, speech and communication, behaviour, transition. Niamh is doing a brilliant job on our Easy Read Guides project, which is, so we're trying to increase accessibility. We've got some lovely My Chromosome and Gene storybooks, which are aimed at children and siblings and maybe friends to explain a little bit about genetic conditions. And we've got an ever-expanding number of translations.